Welcome to Sci-Fi Quantum Archives. Don't forget to like, subscribe, and explore. It began in silence. Not the silence of absence, but the kind that follows precision. A cold, sterile hush filling research corridors and server rooms, where machines hummed not in urgency but in patience. The World Population Genetics Survey had stretched across decades. Quietly funded, minimally publicized, its purpose had never been dramatic. No one expected headlines from a long-term project designed to study the inheritance of chronic disease. They were watching the quiet things. The drift of genes across centuries. The way mutations settled into bone and blood like sediment in the folds of time. In a highland region marked by generational continuity, a community had been selected for its exceptional genetic stillness. Isolation had preserved a lineage largely untouched by migration or disruption. A perfect control group. No war. No exodus. Just repetition. Faces resembling great-grandparents, names circling back through the calendar like old seasons. The third generation was expected to echo the second. No surprises. The sequencing machines processed the samples with quiet confidence, expecting nothing new. But in the data, a pattern surfaced. Not bold. Not loud. Just wrong. Genes that should have followed the laws set down in every biology textbook did not. Traits were appearing in children that had not been present in either parent. Not mutations. These were too specific. Too consistent. It wasn't an error in replication. It was as if the genetic code had made a decision. The lead technician flagged the anomaly. The report was returned with a simple note: rerun the samples. Machines make mistakes. Algorithms drift. Rerun, compare, dismiss. The data was rerun. The anomaly remained. It was dismissed again. Retested in another lab. Same result. In silence, more samples were pulled from storage. Parallel testing. The findings held. It wasn't contamination. It wasn't statistical noise. It wasn't Mendelian. Chromosomes don't improvise. Genes don't vote. Yet this sequence was doing something no inheritance model could account for. It inserted itself with precision. Not random. Not rogue. It followed its own rule, independent of parents, untouched by chance. A senior researcher, someone who had published papers for 30 years without deviation, said the sentence slowly, not to convince others, but to hear it in the air. We thought it was an error in the sequencing machine. But the error repeated. Across multiple sites. Across multiple generations. It behaved as if it had waited. Not for opportunity, but for time. The findings weren't filed away. They were escalated. Quietly. No press. No leaks. This was not a pattern anyone had seen. It was not a variation anyone had expected. It was a signature. And it had been hiding inside the human genome. They isolated it under pressure. Not political. Not institutional. It was the kind of pressure that comes when the numbers refuse to behave. The anomaly wasn't random. It surfaced in multiple lineages, but always with the same behavior. The same placement. The same activation pattern. A segment buried in what had long been called non-coding DNA. Junk, some had once called it. Leftover debris from evolution's long rehearsal. But this fragment wasn't inert. It moved. Quietly. Deliberately. Not mutating, not adapting, not drifting like ordinary sequences do across time. It held its shape. It resisted noise. It corrected damage. In samples showing inherited blood disorders, the damage was gone. Gone, not masked, not diluted by probability. Rewritten. The original mutation overwritten, as if a line of bad code had been replaced by something clean. It had no obvious origin, no homology to any known human, primate, or vertebrate sequence. It did not match archaic hominins. It did not share ancestry with known genetic trait. It was not carried by mutation. It imposed itself. The data showed it appearing in offspring even when neither parent carried it. It was as though it could skip selection entirely. Insert itself with mathematical insistence. Once active, it would propagate not randomly, not passively, but with precision. Not at 50%. Not at 75%. In some cases, it showed up in 93% of viable embryos. As if it wanted to be there. They gave it a name. Not for publication. For reference. The inheritor. Informal. Cautious. They didn't know what it did. Not really. They only knew it didn't stop. They tested for external triggers. Radiation, pollutants, diet, retroviral activation. Nothing correlated. It didn't care about region, age, or environmental pressure. It showed up where it chose to. And once present, it stayed. No decay, no reversion, no sign of being foreign. It nestled inside the genome as if it belonged, but no blueprint accounted for it. The databases were scraped. Every record, every gene bank, every archival file. Nothing. It was not there. It had never been classified. Never referenced in scientific literature. Never mapped. Which meant one of two things. Either it had never existed, or it had always been present, hidden inside the sequences no one ever read, quiet in its dormancy, until something invisible woke it. The ethics committee was contacted in silence. A protocol flagged under genomic anomaly containment. Internal. No statements. No meetings logged. No open correspondence. There was nothing to publish. Nothing that wouldn't unravel trust. They debated classification. Was it a mutation? A regulatory sequence? An ancient genomic parasite? But none of those categories fit. None explained its behavior. None explained why it moved between generations like a directive. Why it spread faster than anything they'd tracked before. Why it corrected instead of corrupted. It should not exist. And yet it was already present in more than a dozen regions, across populations with no genetic link. No migration path. No unifying language. No trade. No war. No shared disease vector. Just the gene. The same gene. Silent for centuries, reactivating as if according to schedule. The World Health Organization was notified. Not through press. Not through channels. Behind closed terminals, encrypted and isolated. The sequence was sent. Secure servers. Triple authentication. Biometric encryption. By the time the confirmation arrived, it had already been sequenced by 3 other labs. In Japan. In Germany. In Nigeria. Each found it without collaboration. Each thought they were the first. They weren't. They weren't even early. It wasn't isolated. The belief that it could be contained to one lineage, one village, one anomaly of chance, collapsed under the weight of new data arriving too fast to track. Laboratories cross-checking sequencing runs began flagging identical structures. Each team thought they were stumbling into something rare. Each flagged it as aberrant, an internal curiosity, not a discovery meant for headlines. But the signature was identical. Not similar. Identical. In lowland Myanmar, a prenatal screening clinic marked an unusually high immunity index in fetuses. In rural Quebec, a hereditary retinal disease reversed in 2 successive generations without intervention. In the outskirts of Córdoba, an experimental gene therapy trial failed to suppress a supposed mutation, but the outcome was a child whose baseline metrics exceeded those of the control population by standard deviations too wide to dismiss. None of these cases should have shared anything. No shared history. No shared exposure. No shared inheritance model. No pattern of admixture. No path for transmission. And yet each sequence matched. Perfectly. It hadn't spread. It had waited. Latent. Dormant. Passive across millennia. Silent inside us. Not selected for, not selected against. Not expressed. Just carried. Something had changed. Not in the sequence. In the world around it. The timeline was reconstructed. Each activation had occurred within the last 11 years. No single event connected them. No common outbreak. No known contaminant. But all samples pointed backward to presence. Deep presence, that predated recorded migration. It had always been there, hiding inside what had once been dismissed as biological noise. The old label, junk DNA, returned like a reprimand. Researchers stopped thinking in terms of origin. They shifted to conditions. What had changed in the human environment to awaken something ancestral? Theories formed, quiet and careful. Radiation exposure thresholds. Trace pharmaceutical accumulation. Soil microbe imbalance. Zoonotic interference. None fit perfectly. None offered a mechanism. But each held a piece. One hypothesis gained traction not for its elegance, but for its simplicity. The trigger might not be biological at all. It might be informational. That the activation was not caused by a chemical switch, but by a density of contact, a statistical weight of genome sequencing. That the act of observing it had changed it. The suggestion was met with silence. Not because it was mystical, because it was deeply inconvenient. A draft paper was prepared by a research group in Sweden. It outlined evidence of the inheritor gene across 21 separate populations, none with shared ancestry within the last 40,000 years. The conclusion was stark, unemotional, and typed without adornment. This is not mutation. This is emergence. The paper passed peer review. The preprint page was created. The embargo date was set. It never published. The submission was pulled 2 hours before release. No reason given. No emails responded to. No author statements. That same week, a deck of presentation slides appeared anonymously on a defunct molecular biology archive. 42 slides, each numbered, each timestamped. The sequence shown at the nucleotide level. Comparative diagrams. Population map. One slide showed activation clusters overlaid with WHO disease alert zones from the last 2 decades. Another showed predicted penetration by 2040, 84%. The forum thread hosting the leak was deleted after 4 hours. Not before it had been downloaded hundreds of times. The data could not be contained. Not because it was public. Because it was already inside the people reading it. They ran the models again. Every simulation, every variant, every known reproductive pattern across all documented species. Nothing aligned. It didn't follow survival pressure. It didn't act in response to external stimuli. It operated beneath both mutation and selection, beneath struggle, beneath death. The gene ignored the core assumptions of Darwinian biology. It did not wait for the environment to punish weakness or reward advantage. It moved on its own, as if carrying a blueprint no one could read. It selected. Not for strength. Not for adaptability. For something else. Something structural. It reinforced neurological repair over aggression. It favored intergenerational communication over reproductive speed. It didn't optimize for the present. It shaped lineages as if the body were only scaffolding and the inheritance the real structure. Traits were passed that had no survival benefit in the wild. They conferred no reproductive edge. They offered no resistance to disease. But they repeated with precision. Memory retention. Recursive reasoning. Anomalously high pattern sensitivity in early developmental stages. Infants that stared too long. Children who spoke late but never forgot what they heard. This was not a shift. It was a direction. It broke the premise that evolution was blind. Broke it quietly, clinically, without fanfare. The idea that nature selects randomly from noise had never been challenged like this. Variability had always been the engine. Competition the filter. But this was neither. It was not chaos winnowed by time. It was intention. A long silence followed. In research institutions, philosophers were called into rooms they were never invited to. Ethicists asked questions they had stopped asking decades ago. The arguments restarted. Teleology, group selection, holism, concepts that had been discarded as metaphysical, unscientific, rhetorical fossils. Comparisons were made. Transposons, those selfish pieces of code that copied themselves across the genome. But this wasn't selfish. It didn't replicate blindly. It inserted with restraint. always in the same location, always with the same effect. Group selection was suggested, traits passed for the benefit of the population, not the individual. But no group defined here benefited directly. The effects span timeframes beyond any natural system's feedback loop. There was no reward in one generation, only direction. Every model collapsed under the same failure. This gene acted as if it had knowledge of what it was doing. No protein does that. No sequence anticipates. But this one corrected, repaired, removed errors not in function but in form, not in sickness but in deviation. There was no predator to select against it. There was no disaster to reward it. It simply rewrote what had been broken. Silently, reliably, without mutation. The first conference to address it was cancelled before it was announced. The paper that tried to quantify it never passed review. The terminology lacked grounding. They had no precedent. No theory could hold what this gene did without unseating what biology claimed to know about life itself. The question was not where it came from. That had become irrelevant. The question was why it waited. The suggestion emerged slowly, carried not by data but by discomfort. A conclusion no one wanted to say in full. What if evolution wasn't finished? What if the randomness we had mapped was only the scaffolding for something else, something waiting for its conditions to return? What if we were not the product but the process? And what if that process had just resumed? Across the globe, in samples arriving unrequested, the same sequence began appearing with greater frequency. In cities, in refugee camp, in fertility clinic, in populations never sequenced before. A curve began to form. Not exponential, deliberate. The numbers were clean. That was the part no one could explain away. In 3 years, average gestation outcomes among carriers improved by measurable margins. Infant mortality among previously high-risk demographics dropped below statistical floor. Neural imaging in early childhood began revealing anomalous symmetry in regions once considered chaotic. Immunoresponse to chronic pathogens stabilized in patterns that didn't match vaccine history. Children born with the sequence showed predictive cognition years ahead of baseline. Not genius, not gifted, just precise. A kind of unspoken accuracy. They asked fewer questions but saw more. Hospitals stopped calling it an anomaly. Insurance forms adjusted. Guidelines revised without announcement. Medical boards met quietly, out of rhythm with standard cycles. The change was not announced. It was registered. Attempts to contain it failed before they began. Airborne markers showed up in unrelated regions. Waterborne DNA fragments traced upstream from uninfected populations. Blood donations from carriers carried no symptoms, no instability, no illness. There was no vector to isolate. The mechanism of spread was not mechanical. It was reproductive. Carriers passed it not by chance, but by inevitability. It didn't behave like a pathogen. It wasn't fought like one. But the policies were the same. Border screenings, quarantine zones, discrete travel bans, genomic screening added to visa applications under vague public health pretexts. What began as scientific caution turned bureaucratic, then political, then religious. There was no unified response. In one region, church congregations baptized carriers as harbingers of restoration. In another, excommunications were issued before the genome reports had even arrived. Faith fractured along lines that had nothing to do with doctrine. Across denominations, the divide was simple. Either the sequence was salvation, or it was interference. It spread faster among those who embraced it. Within months, underground clinics began modifying embryos, not correcting for disease, not enhancing for intelligence, just inserting one silent gene, one line of code that had already begun asserting itself across the world. No one regulated it. No one could. The procedures were cheap, the tech already old, and the demand immediate. No one sold advantage. They sold inclusion. Children born from those clinics were called many things. In official records, they were indistinguishable. In whispered conversations, they were marked. A new movement formed. Not in the street. Not on official channels. It grew where surveillance never bothered to look. Among teachers who saw changes they couldn't explain. Among parents whose children no longer fit the expectations they'd been taught to hold. Among midwives, data technicians, retired physicians. People who noticed. People who stopped resisting the pattern. They called themselves the heirs of the code. No founding document. No leader. No doctrine. Just a belief passed like breath. The gene was not intrusion. It was return. It was restoration of what had been paused, misdirected, silenced. They stopped asking where it came from. They started asking what it was making. Public reaction fractured faster than institutions could adapt. France saw 3 nights of fires outside a genetic screening center after reports leaked of infants denied coverage based on unknown carrier status. In Sao Paulo, a clinic offering elective insertion of the sequence was destroyed in a coordinated attack that left no survivors. In Jakarta, genome testing kits were banned without explanation, the sale of private diagnostics criminalized under public health orders. Social media purges failed to erase the footage. A child standing in a school hallway, eyes fixed not on a teacher, but on the oscillating pattern of the lights above. A woman holding a printout with an empty diagnostic field. No anomaly detected, but the system refusing to flag the result as normal. A man whispering on a metro platform, not preaching, not persuading, just repeating 4 words. It is already written. There was no single narrative. There was only a question no one could word without forfeiting what they believed humanity had always been. What happens when biology stops waiting for permission? An agreement was drafted across closed doors and encrypted lines, not to solve the crisis, but to delay its consequences. The inheritor gene, once treated as anomaly, had become a line item in international law. No intentional editing. No distribution. No insertion beyond natural inheritance. Not because it could be stopped, but because something had to be written before control was entirely surrendered. The signatures were ceremonial. The signatories, aware of their own redundancy, complied for appearance. The sequence was already beyond them. Enforcement faltered before implementation. Documents circulated, first restricted, then leaked, showing classified trials. Not in clinics. Not in research labs. In controlled combat units. Soldiers whose wounds closed without scarring. Soldiers whose reaction times were no longer bound by adrenaline. Units that moved without hesitation, not out of obedience, but out of clarity. Reports described cognition stripped of interference. No panic. No indecision. The term used was clean. The programs were unofficial, unconfirmed, unacknowledged, but they were not unique. Signals intercepted by intelligence analysts suggested another country had stopped identifying carriers. Not to track them, to recruit them. A pattern formed in border regions where carrier populations were rising. Not through medical necessity, through coordinated placement. Cities restructured their schooling systems to isolate those testing positive. Not for containment, for specialization. By the time the briefings reached diplomatic channels, the data was too widespread to challenge. Carrier groups were being armed, trained, structured under civilian cover. The speed at which these deployments had taken place indicated foreknowledge. The gene hadn't been discovered by one institution. It had been identified by many, but only some had chosen to keep quiet. Scientific journals fractured. What had once been consensus dissolved into contradiction. One editorial board published a study claiming the sequence was a dormant viral remnant reactivated by pollution threshold. Another published a rebuttal signed by 16 independent labs, asserting the data had been falsified. A third published both, side by side, with no comment. Careers ended in silence. Credentials stripped. Labs defunded. Others surged in influence, not for rigor, but for alignment. Research conferences became battlegrounds, not for facts, for control over what counted as truth. An open letter was circulated across institutions in 48 countries. It carried no hypothesis, no agenda, no conclusion. Just a single line. We no longer know what it means to be human. The message wasn't rejected. It was absorbed. It matched what many had already begun to suspect. Identity, once grounded in biology, had begun to loosen. Citizenship, language, borders, even memory. None of it mattered to a gene that cared only for replication on its own terms. Parents reported children speaking sentences that hadn't been taught, languages no one had spoken in their homes. Infants responding to rhythms with eerie precision. Early dream patterns recorded during sleep studies began clustering. Recurring shape, recurring geometries, patterns too specific to be symbolic, too consistent to be accidental. The content of these dreams was not frightening, not violent, but the detail was impossible. Children waking up with numbers, grids, ratios, not imagined, not babbled, drawn. Something was unfolding in cognition, not imagined intelligence, not genius, something else, an echo in the structure of how thought began. Something familiar to no one, and identical across those who had never met. It wasn't evolution. It was instruction. It began without language. Not silence, something beneath it. The quiet not of absence, but of attention forming without sound. Carriers, observed closely, began to act before being asked. Not in learned routines, not in rehearsed responses, but in sequences too precise to be accidental. Objects found before being requested. Faces remembered after a single glance. Routes traced through unfamiliar places with no correction. An emergent intelligence without articulation. Memory detached from rehearsal. Reaction decoupled from input. Not faster, not sharper, more certain. Like the space between thought and action had shortened without compression. Tasks completed before explanation. Decisions made without deliberation. Not instinct. Not reflex. Something else. Quiet precision without urgency. Educators noticed it first. Children no longer stumbling through mistakes, no trial and error. They paused, observed, acted. Not obedient. Not distracted. Present in ways that couldn't be trained. Symbols began appearing. Not taught. Not prompted. Drawn without context. Circles within circles. Lines intersecting at irrational ratios. Shapes folding in on themselves while retaining symmetry. The same forms repeated across continent. In chalk on pavement. In condensation on windows. In margins of school notebooks where no curriculum existed. Symbols seen by no one before. But drawn by many. They were not decorative. They were not random. Analysts from independent systems reported properties buried in the ratios, mathematical identities embedded in shape. The patterns carried recurrence, not repetition. Fractals that unfolded with internal constraints. Recursive forms that respected limits no child had reason to know. None of the children could explain them. They didn't try. Neuroscientists began recording sleep cycles in controlled environment. REM phases showed patterns of neural activity never previously documented. Not louder, not stronger, aligned. Groups of children placed in separate chambers showed synchronous spikes without common sensory input. No environmental trigger. No visible stimulus. Entire clusters of neurons firing in spatial coordination with subjects miles apart. It wasn't telepathy. No thoughts transmitted. No voices shared. But something was forming across those carriers without conversation. Information without signal. Organization without command. The sequences didn't behave like enhancement. No acceleration of learning. No transfer of skills. But recognition arrived earlier. Patterns seen before they were identified. Structures understood before being named. Behavior changed not by pressure but by clarity. Answers no longer guessed, just known. Cognitive infrastructure. That was the term used by the teams that first compared the scans. The gene wasn't upgrading the brain. It was arranging it, wiring pathways that anticipated function before experience. Not for control, for readiness. The inheritor had never been passive. Not a passenger in the genome. Not biological drift. An architecture waiting for completion. It hadn't added something new. It had revealed something withheld. Across the world, adults began noticing they were no longer the first to understand. The questions asked by the next generation were no longer naive. They were architectural. Not what is that, but why is it arranged that way? Not who made this, but what was it made to become? The shape of thought was changing. Something had been reconnected. Not taught. Not conditioned. Restored. Instruction without instruction. Pattern without imitation. Understanding without explanation. A signal that had always been embedded, waiting for the circuitry to activate. No transmission. No discovery. Just recognition. They do not reproduce as their parents did. Not through rejection or resistance. Not through rebellion or refusal. Through redefinition. Biology reshapes itself without permission. Not abrupt. Not declared. Just revised. The markers are clear. Sexual differentiation no longer aligns with traditional developmental phases. Internal hormonal cycles shift without pharmacological input. Traits once tied to external gender markers emerge independently, selectively, rhythmically. Fertility windows close later. Puberty begins earlier. Longevity curves extend without intervention. Medical baselines recalibrate. The averages no longer reflect the species once mapped. They belong to something else. The gene has touched the feedback loops that govern response, memory, perception. The old boundaries that define self from other, mother from daughter, brother from stranger, memory from instinct, begin to soften. Identity no longer solidifies along the same coordinate. It becomes fluid not in surface expression but in structure. Carriers do not simply feel different. They are language fragments in their mouths, not because of trauma or confusion, but because it no longer serves the same function. Words fail to capture what is now observed intuitively. Vocabulary is outpaced by recognition. Communication shifts toward economy. Short phrases, direct expressions, proximity replaces dialogue. Gaze replaces negotiation. Some still speak, but less often. Not from silence, but from sufficiency. The old terms collapse. Intelligence, gender, family, nation. Their definitions dissolve not from ideology, but from irrelevance. The boundaries they once enforced carry no tension in the new physiology. There is no resistance, only substitution. Those who remain unchanged feel the difference before they can describe it. Not outcast, not excluded, not shunned, simply bypassed. Their pace no longer matches. Their rhythms no longer sync. Laughter no longer lands in unison. Shared references begin to thin. Meals feel longer. Conversations close more quickly. Eye contact lingers too long or not at all. They were not targeted. They were not diminished. They were not erased. They were simply no longer aligned. A boy asks his grandfather why people used to speak with their mouths more. The answer takes too long. The boy is already gone, chasing a shape in his mind he doesn't yet know how to draw. The last non-carrier generation watches the world continue without waiting. They are not bitter. They remember when it was theirs. They remember how it felt to define the future. But that sensation has drifted, quiet and unannounced, like breath cooling against glass. Medical reports cease comparing metrics to historical baselines. The past no longer offers meaningful context. The charts begin with the current generation. The rest are placed in archives. The separation is not political. It is not legal. It is not enforced. It is biological. They are no longer the same species. No one holds a ceremony for it. No treaties mark the threshold. But it passes, quiet as nightfall. A moment no one names because naming belongs to an earlier pattern of thought. For centuries, humanity believed it stood at the apex of a random climb, a product of chance and survival. We thought we were the result. Maybe we were the interval. Maybe we were just the chrysalis. Evolution has ended. Not with collapse. Not with extinction. With a correction. Inheritance no longer drifts. It no longer waits for error, punishment, chance. It proceeds with intention. The engine that once churned through randomness has gone still. Not broken. Retired. Where variation once scattered across generations like weather, precision has taken root. Traits are no longer gambled. They are placed. Not engineered by human hands. Not directed by policy or preference. The sequence does not ask. It implements. The change is structural, quiet, complete. The world did not burn. Cities still breathe. Seasons still turn. Grief still exists. Love has not vanished. But the way choices are formed has shifted. The source of difference has moved from the surface to the design. The shape of thought, once built by accident, is now woven with purpose. Most do not notice. Children speak in shorter phrases. Their eyes settle differently. They look at problems not to solve them, but to complete them. People gather with less noise. Music shifts toward intervals that reflect something unlearned. Stories become less about conflict, more about recognition. No one taught this. No one requested it. It arrived through blood and breath. Hospitals have grown quieter. The need for intervention has declined. Certain procedures are no longer performed because the conditions they once addressed no longer appear. Schedules shift. Care becomes observation. Monitors rarely sound. In one room, at the edge of a sleeping city, a man breathes slowly beneath a dim light. His records show no sequence. He belongs to the last registered cohort without the gene. No announcement was made. No obituary written. The data will mark his passing, but the world will not stop. It does not need to. He is not afraid. He has watched the change. He has seen what has taken his place. Not a replacement. A continuation. The lights above his bed pulse in rhythm with the equipment beside him. It will be the last time this calibration is required. 4 billions of years life survived by chance. One gene changed that. It didn't make us gods. It made us deliberate. Thank you for watching and sticking with me to the end. Now, if you believe humanity deserves a future among the stars, do subscribe now. Only those who'd abandon Earth to its fate will scroll past.